Hello and welcome to viruses. We're going to dig a little bit deeper into this fascinating subject, and at the end of today's lectures, you should be able to know how viruses are cultivated and assayed. You should be able to distinguish the 7 different types of viral genomes and their structures, and you should appreciate what information is and is not encoded in a viral genome. Let's first start by talking a little bit about how we study viruses. Now, you remember from our introductory lecture that animal viruses were discovered around the end of the 1800s. But for many years, these viruses could not be routinely studied in cells in culture. They had to be studied in laboratory animals, and laboratory animals of all sorts were used. Some of them are shown here. But as you can imagine, this is not a convenient procedure. Plus, laboratory animals can differ from experiment to experiment. So it was very important to try and determine how to grow viruses in cells, and many scientists worked on that problem. Before 1949, it was not possible to use cells in a consistent and reproducible way to study viruses. But in that year, 3 scientists, Enders, Weller, and Robbins, working in the US, found that they could propagate poliovirus, a human virus, in human cell cultures. Now, they happened to use what we call primary cultures from embryonic tissues, but this experiment set a precedent. For the first time, it was possible to propagate a human virus in cell cultures. This was a very important discovery for which these researchers received the Nobel Prize in Medicine in 1954. That discovery is with us today. Since 1949, we've been able to study viruses in a variety of cell cultures derived from different sources. For example, we can use primary cell cultures, where the tissue is taken from a source, it's minced up to produce individual cells, and then it's put on a plastic layer in a dish and covered with medium. So on the left-hand part of this slide, you can see a photograph of primary human foreskin fibroblast cells in culture. Human foreskins are very easy to obtain because when babies are born, we often remove the foreskin. It's thrown away in the hospital. But if you're interested, you can obtain them and produce primary cells like these. This, of course, is not so convenient to use on a routine basis. So for that reason, we have what we call cell lines. And there are 2 examples of these cell lines shown on this slide. One is a mouse fibroblast cell line. It's called 3T3. And then there is a human epithelial cell line, the famous HeLa cells. Both of these cells are what we call immortal. They grow forever. You can grow them over and over in the laboratory, and then that makes it a lot easier to do your experiments. Now, the negative aspect of using a cell line is that they're a little bit unusual. They have far too many chromosomes, they're a bit abnormal, and some of them are, in fact, cancer cells. So if this is a problem for your research, we also have what we call diploid cell lines, like the WI38 cell line, which is derived from human embryonic lung. These have a normal number of chromosomes. They don't live forever, but they do live a long enough time to make your experiments convenient.